All right, let's talk about lab 10, which is about blood typing. I just want to go over, I just want to extend a little bit what some of the background information says about blood type and antibodies versus antigens, because I feel like with both of those words starting with anti, it can get a little bit confusing. So I have a PowerPoint that I want to show you that should help clear up any confusion you might be having. All right, so for the most part, there are 4 different blood groups. There are a few other types of blood that are rare, really rare, but we're going to talk about just the 4 that most people know. Most people, if you know your blood type, you know you're either A, B, or O. And then you may have a positive or a negative. I'm going to talk about the letters first, and then the next slide we'll talk about whether you're positive or negative. Basically, what happens is every single cell in your body is covered with what are called antigens. They are tiny little proteins that kind of stick out from the cell membrane. They are what the body uses. They can signal other cells with them. They are ways that the body can tell if that cell actually belongs in your body or if it is foreign. They have lots of different purposes. They do various different things. So I'm only going to talk about them in terms of blood type, though. What happens with blood type is you can have a possibility of A antigens, B antigens, A and B antigens, or no antigens at all. In fact, O comes from 0. In other countries outside the United States, sometimes they call it 0 blood. Because what happens is on the outside of a cell, if this was a regular blood cell, you have all these little proteins called antigens that are there. There are like thousands of different kinds, and this is just one type for blood. So if you have what are called A antigens, you have this protein that is in this specific shape. It is not really a circle, but we are using that for the purposes of us being able to recognize that there are differences. So A antibody, A blood, you've got these A antigens. These are hanging out on the outside of your cell. It tells your body, hey, I belong here. This is what's going on. If you have B antigens, these are in the shape of a little diamond. It's just a different shape. It's a protein with a different shape and maybe a slightly different function. But for the most part, as far as blood type is concerned, it just helps your body know it belongs here. If you have AB, you have both A antigens represented by the little circles and B antigens represented by the little diamonds. If you have O type blood, you have none of these antigens on your blood cells. So you do have other proteins, you do have other antigens, you just don't have A or B. So that's all fine and dandy. The problem is, especially when you're talking about blood transfusions or blood being contaminated or anything like that. The problem is if you have A antigens, so your blood cells have this protein labeled A, your body, your immune system will make antibodies against the B antigen. And the way antibodies work is they have the short version of this, you can learn this better in other classes, but the short version is they have these chains that come up and at the ends of them will form different shapes. And these antibodies will go and try to match antigens on cells. You've got antibodies flowing around in your bloodstream all the time that are just going around and they check on cells and they check on things. They find something that matches. That means that it's a foreign body and needs to be stopped. So once your antibodies find something that they match, if it matches, it means it does not belong in your body. So it will match it and it will instigate an immune response. And anytime your body instigates an immune response, it calls everybody in. It's like there are all kinds of cells in your immune system that will come in and they'll start to attack that to make sure that that's what helps keep you healthy. So if you have a antigens on your blood, then you make anti-B antibodies. So if you got B blood show up in your system, then your anti-B antibodies would attack that B blood, call in all the cells it can, and it basically forms clots and problems. It agglutinates, meaning basically that it like clumps up. Well, when you think about your blood vessels, they only have so much space in them for your blood to flow through. And you think about capillaries that are so tiny that one blood cell is all that can fit through and even has to squish. If you start getting things attacking and attaching, you're going to end up with all kinds of blood clots. You can end up bursting blood vessels, especially like the thinner capillaries and stuff. You can have all kinds of problems. So this is why knowing your blood type is important. And when we do transfusions, it's really important to make sure you match the A and the B. So if you have A antigens, you make anti-B antibodies. If you have B antigens, you make anti-A antibodies, so that if you were to get some A type blood in your body would react. It would start an immune response. If you have both A and B antigens, though, you don't make any antibodies against it. So you could get blood from someone who has A blood, someone who has B blood, or you could get it from someone who has O blood, because there's no antigens on it, no antibodies to attack those antigens. If you have O blood, though, you make both anti-A, anti-B, and anti-B antibodies. So if you were to get any A blood, B blood, or AB blood in your system, you would trigger an immune response. So this is why blood type is important, and this is what it means. So when you are testing for blood type, you will have a little plate, and this is what you're going to be doing in this lab. You'll have a little plate, and you'll put a drop of anti-A antibodies into the little well labeled A. You'll put some anti-B antibodies into the little well labeled B. And then I'm about to talk about the positive and negative. That's another antigen altogether that you'll put in that well. And then when you put the drops of blood in, you'll mix it up. And if you get agglutination or clumping, if you prefer that word, then you will know that that is the type of blood you have, because the anti-A antibodies reacted with those A antigens. All right. Rh factor, this is where the positive and negative comes in. For instance, I have O positive blood. That means that I have no A or B antigens. My body does produce anti-A and anti-B antibodies, and I do have the D antigen, which is the Rh factor. So D antigen, this one's separate from the A and B, but it's pretty important. If you have the D antigen, you could have either of these genotypes, which that previous chart talked about phenotypes versus genotypes. I'm not going into all that right now, but it is dominant. You can see that with this genotype. So you do not have any antibodies against D, because then your body would be attacking itself. So that doesn't make sense. So you don't have, I'm O positive, I have the D antigen, and I do not make antibodies against it. If you have a negative type blood, doesn't matter what the letter is, because those are separate from this. But if you have a negative blood, then you do not have the D antigen on your red blood cells. You are considered Rh negative. It is recessive, so your genotype is going to be homozygous recessive. And then you only produce antibodies against D blood if you're exposed to it. So for the most part, it's very rare to have someone else's blood put into you unless you are, unless you're having a transfusion. But your body does not start producing the antibodies against it until it's exposed. So the biggest problem where this can occur is in hemolytic disease of the newborn. And it is because during birth, if a mom is Rh negative, does not have the D antigens, and the father of her baby is Rh positive, and that baby is positive, they're fine, usually fine through the pregnancy for the first one. But during birth, mom's blood and baby's blood can get a little mixed up, and the body can be exposed to that D antigen and start to produce antibodies. So the problem becomes in the second and third and fourth and subsequent births. It's explained really well on page 3 in your background information of this lab. So that's about all I'm going to say about that. But that's something to definitely be aware of. So also on page 4 of your lab, you have a really good chart that helps you know if this blood interacts with this antigen, or if it has this antigen, it will interact with this antibody. And helps you remember what you've got if you're different blood types. Anyway, so read that chart. But when you do your blood typing today, this is a sample of a blood type. So what happens is you've got the agglutination. So sometimes it looks like this, and you can definitely see tiny little, like, it's like the blood kind of like goes liquidy, and then you get all these little red clumps in it. That is what agglutination often looks like. This one, it just looks a little bit watered down. So they put they put the blood in, they added some anti-B antibodies, and it just kind of watered it down. It didn't really do anything. And then with the anti-D, you get that agglutination again. So this is an A positive because you got agglutination with anti-A antibodies, meaning the anti-A attached to the A antigens on that blood. So it means that blood has those A antigens, and also your anti-D antibodies attached to the D antigens on the that is on that blood. So this is how you get an A positive. Now, I will warn you, you are using synthetic blood and synthetic antibodies in your kit. So sometimes the agglutination does not look exactly like this. Sometimes it does, and it's very clear that it's like something separated and it's just weird. It doesn't just look watery. Occasionally, though, I've used this synthetic blood and these synthetic antibodies many times in many labs. And occasionally, what will happen is it clumps up, but it looks like this. And so if you let it sit for a couple minutes and then you poke it with your toothpick, it's like this jello-y nasty mass. So be sure before you read your antibody testing, your blood type testing, be sure you check it after a couple minutes to see if it's still just, all liquidy and fine, or if it's separated, or if it's gone all clumpy and jello-like. And that will help make your tests easier to read. So as always, if you have any questions, feel free to reach out to me or our TA during office hours or by email. This one should be a pretty quick lab to do. Make sure you read the background information, and hopefully the pictures and the charts that I've got in here, in addition to what you've got in the lab, will help kind of clear up exactly what they mean by the anti-A and anti-B versus the antigens. So good luck, guys.